Hi everyone, my name is Diana Delgado, and the microorganism that I chose is Plasmodium falciparum and is also known as malaria. The article title is Genetic Variations of Plasmodium falciparum circumporozoite protein and the Impact on Interactions with Human Immunoproteins and Malaria Vaccine Efficacy. The authors are Cheek Campbell Ding, Kobe T. Ford, Anita Lurk, Dixon Deneau, Kovid Vagensa, Daniel Janies, Li Wang Kui, Linda Amoa, Ya Afraid, and Eugenia Lowe. The article took place at the University of North Carolina, University of Notre Dame, University of Ghana, and the University of South Florida. This is the first page of my article. It has the title of the journal, the title of the article, and the abstract, and a little bit of the introduction. In the background section, it talks about how malaria has caused over 219 million clinical cases and 435,000 deaths. In 2021, the first world's malaria vaccine was endorsed, even though there was a low efficacy against Plasmodium falciparum. Throughout the studies, they will determine multiplicity and CSP diversity of P. falciparum. CSP also stands for competent stimulating peptide. The author's hypothesis is that a longer peptide of the Th2R, Th3R CSP regions needs to be considered in future vaccinations. Th2R, Th3R are T helper cells that help with your immune system. The methods are that samples were collected from 5 different sites as well as 88 capillary blood samples from subjects who were diagnosed positive in malaria. They did a PFCSP sequence analysis, and PFCSP stands for Plasmodium falciparum circumporozoite protein, and they had a haplotype network, which is used to visualize relationships among DNA sequences within a population or species. The haplotype network was generated with TCS, which stands for two component system, and then the library was used to obtain TCS scores. All sequences were analyzed together and a phylogenetic tree was reconstructed. They generated CSP peptide structures and the reference structure with HLA-2A PBD using haddock. HLA-2A is a human leukocyte antigen, and haddock stands for high ambiguity driven protein docking, which is an information driven flexible docking approach for the modeling of biomolecular complexes. The haddock scores change from different types of energies. They did measurements of anti-CSP antibody levels. They used recombinant IgG, which is a type of antibody, for standard calibrator of total IgG measurements. In the results for the MOI and haplotype variations, and MOI stands for multiplicity of infections, there were a total of 30 haplotypes that were detected, and 80% of the samples polyclonal infections were detected. For interactions for HLA alleles and PFCSP peptides, 30 HLA class 1 molecules were able to bind to PFCSP haplotypes. 55% showed a higher binding affinity than PFCSP reference 3D7. 3D7 is also a type of antibody for P. falciparum. The association of PFCSP antibody levels with ligand peptide binding affinity were measured. CSP antibody level was associated with CSP peptide with HLA, but it was not associated with qPCR based parasitemia among infected samples. Based on haddock, the binding peptide for Th2R ranged 4 to 9 residues in length. Longer peptides showed a higher binding affinity with the HLA groove. In the graph, the x-axis is the peptide molecular weight, and the y-axis is the haddock score of HLA-CSP peptide. It has a strong negative correlation between both of them, suggesting that a longer CSP peptide confers binding affinity. In conclusion, PFCSP haplotypes are not related to 3D7. The length of Th2R peptide is correlated with binding affinity, but 3D7 does not impact binding interactions, causing short CSP peptides. These findings may only represent only a portion of CSP variants present in Africa. The scientists suggest that there needs to be a longer CSP peptide for the next generation of RTSS for high vaccine efficacy. Thank you, and that is it.